We're really excited to have a joint American Thoracic Society and Japanese Respiratory Society webinar today, presented by G&G and RCMB. And so the talk will be Frontiers in Omics, Translational Insights from Japanese Respiratory Society and American Thoracic Society experts. So it's 6 p.m. in Eastern Standard Time. It's actually 8 a.m. in Japan. So we thought that was really exciting. We wanted to say good morning to Japan, or ohayo gozaimasu. Good morning to you. And so first I'll be introducing our amazing speakers and our moderators. And then we will have our talk from Dr. Bowler. And then following that, we have our talk from Dr. Edahiro. And then we will have a panel discussion and Q&A. And so if you would like to ask a question during the webinar, please put your questions in the Q&A. And then at the end, you can also raise your hand if you'd like to speak and ask your question directly. So I'd also, well, first, I also like to introduce our moderators. So my name is Dr. Amy Attaway. I am from Cleveland Clinic. And then I have Dr. Merry-Lynn McDonald from the University of Alabama at Birmingham. And then also we have Dr. Shimpei Gotoh, who is from Kyoto University. He is also one of our moderators. And we'd like to thank from the Japanese Respiratory Society. And then, so I think why don't we start our presentation? So our first presentation will be from Dr. Bowler at Cleveland Clinic, and he will be presenting on integrating multi-omics to understand COPD pathogenesis.
Okay, great. Thank you very much. Thank you, Amy, for introducing me. I'm very excited to have this joint webinar. It's something we've sort of realized doing respiratory genomics and multi-omics, that it's really important to have international collaborations and international representation in studies. So today I'll be talking about integrating multi-omics and to understand the COPD pathogenesis. As Amy mentioned, I'm Russ Bowler. I'm at Cleveland Clinic. And for those of you who don't know where Cleveland is, Cleveland is in the eastern central part of the United States along the Great Lakes, which are shown here. And we're right in the middle of one of the more shallow Great Lakes, Lake Erie. And then you can see where my lab is circled in red. All right. So I have no significant conflicts of interest for this talk. And really would like to just share some of the people who've done this work and a lot of the funding industries, including the National Institutes of Health, Department of Defense, and some other grant funding resources that have supported this research. We will be talking about COPD, and I'll have a brief paradigm on why this might be a good disease for studying complex and heterogeneous diseases. We'll talk a little bit about the public health significance of COPD, and then I'm really going to present two strategies. So I've been doing science for over 30 years now. I actually published my first paper about 40 years ago. And when I started, it was in the pre-omics era, and we really focused on candidate genes and proteins. And as about 25 years ago, with the advent of sequencing and the whole genome sequencing of humans, we've sort of entered this omics era, and now I would say the post-omics era. So we'll talk about both of those, contrast them, and I'll give you examples of omics discoveries that we've made in some of our COPD studies. In particular, we'll talk a little bit about ECSOD, which was a pre-omics era discovery, and a protein called RAGE, which is really come about in the post-omics era. So just a quick background. We don't have a lot of time, so I'm not going to spend too much on this, but COPD, as many of you know, is a disease that's been increasing in prevalence over the decades. It closely is related to tobacco smoking and typically follows peak tobacco smoking by 10 to 20 years. It is the only major disease in many Western countries, well-developed countries, where the mortality has been increasing. Unlike other diseases such as cardiovascular disease and strokes, which have been, at least in the United States, have been decreasing in both mortality rates and prevalence. So the neat thing and difficult thing about COPD is that everybody is born, for the most part, with healthy lungs. They're very white, and for those of you who do bronchoscopies, you'll note the difference between a young person's lung and an old person's lung, and that in particular, those who smoke or live in high pollution areas, their lungs start to become dirty and their lymph nodes along their airways are quite visible and dark. So really the question in COPD is why do people develop this? So the majority of people who smoke actually have pretty normal lungs, at least by spirometry. So really the question is, why do some people develop lung disease and others don't when they smoke? And that's what we'll talk about today. And then there's some secondary questions about smoking. In particular, why do some people develop different phenotypes of COPD, such as airflow obstruction, emphysema, or chronic bronchitis? Just a quick review. I mean, you're all pulmonologists, so it'll be very quick. Typically, COPD is defined by spirometry, and it is defined by a post-bronchodilator obstructive lung disease, as shown here. So there's impairment in airflow outside of the out of the lung, particularly at 1 second, and that's how we stage it. But more recently, I think some of the work we've done in COPD gene and spiromics have helped establish that emphysema is another phenotype of COPD, and we've developed quantitative measures of emphysema by CT scans. Currently, most of those quantitative measures are density-based, so really leveraging the difference between air, which is low density and typically close to minus 1,000 Hounsfield units, and lung tissue, which we like to say is sort of a more dense, and that's above 950 Hounsfield units. So a typical way of quantitating emphysema is to take a CT scan and mask out all the non-lung parenchyma tissue, and then quantitate the density of pixels or voxels. And what we'll often do is assign a voxel that's less than negative 950 Hounsfield units as an emphysema voxel. Another way of measuring is emphysema is to look at the 15th percentile of voxel density. So this is a way we quantitate emphysema. A lot of this has been pioneered in COPD gene spiromics. And then, of course, there's a clinical definition of chronic bronchitis and COPD exacerbations. And that is characterized by lots of airway inflammation, hypertrophy, mucus secretion, et cetera. But I think the really important thing to learn about COPD is that all of these phenotypes, emphysema, chronic bronchitis, airflow obstruction, they're quite heterogeneous in a population of smokers. This graph from COPD gene includes all 10,000 people that were enrolled. And I've plotted on the x-axis airflow obstruction, so everything to the left of the line reflects an airflow obstruction. And also on the y-axis, I've plotted emphysema, so everything above the line above the 5%, which we've given as a normal level in the lung. So everything above that is, we would say, people have emphysema, and I've color-coded the dots on whether or not they have complaints of chronic bronchitis. The point of this graph is really to show you that COPD is heterogeneous. Some people can have different amounts of emphysema versus airflow obstruction and chronic bronchitis. And that we can leverage that heterogeneity in our studies. All right, so first thing I'll talk about is what was multi-omics and what was COPD, how was it studied in the pre-omics era? And this was before we had whole genome sequencing and genotyping arrays, we would typically target particular genes and study them. So I did my PhD in cell biology and was very interested in antioxidants. And 20 to 30 years ago, some of the leading pathogenesis theories of COPD was that smoke caused excessive oxidative stress on the lung epithelium, and it overwhelmed antioxidant defenses. And so I studied animal models in the lab of antioxidants. And one antioxidant in particular that we were very interested in is the most abundant protein, so enzymatic antioxidant in the lung, which is called extracellular superoxide dismutase. And you can see that it is highly expressed in lung epithelium, in the submucosal area of the airways, and then also in blood vessels within the lung. So one of the things we noticed is one of the first things, and I'm not going to present too much data, that I did was collected several hundred COPD gene subjects' blood, and I wanted to look at genetics to see if there were any genotypes that were associated with COPD. And we did this with J. Craig Ventnor, who was really the founder of Celera, and he was the first person to sequence the human genome. And we did this right after he finished. He had lots of excess sequencing machines available. This was about 25 years ago. And I brought in these samples and we sequenced this SOD3 gene. We found lots of mutations, and then I worked with collaborators in Denmark and also cohorts in the United States. And we found one very interesting mutation in the SOD3 gene, which codes for the protein called ECSOD. And this polymorphism, which we call the R213G polymorphism, this is a missense mutation. It was associated with protection from COPD. This is in the Copenhagen City Heart Study. So I was doing my PhD and very interested in proteins. So I wanted to figure out what did this protein do. It was a missense mutation, as I mentioned, and it had this particular feature that we found important. It caused a change in an arginine, which is a positively charged amino acid, to a glycine in a region of the protein, the carboxy terminus, that was very important for binding to the extracellular matrix. So it disrupted this. Then we want to ask ourselves, why was this causing COPD? What was the mechanism? And this is where knowledge of genetics came in, is that we quickly realized that the human, which is shown on the top here, that sequence of the protein and also genome was nearly identical to mice. And so we decided to make a knock-in mouse. This was before CRISPR-Cas, where we could knock in the exact same missense polymorphism to basically replicate the polymorphism in mice. And this was done, again, before CRISPR-Cas. Now it's straightforward to do this. It took about 2 years to make that mouse. So we made the mouse, and one of the things we learned was that this polymorphism caused a shift in the protein from highly bound to lung tissue, intracellular or extracellularly along the matrix. And when you had this polymorphism, it disrupted the binding to collagen and heparin and resulted in the protein redistributing from the lung tissue, as shown on the left, into the BALF, which is shown on the bottom panel. So you get a shift from the protein of lung tissue into the BALF. There was no change in the enzymatic activity of the protein, just a distribution shift. And we saw that both in the mouse that we made and in the humans that we studied. The result of this shift was an accumulation of SOD3 activity, so this antioxidant in the airways, in particular not shown, but in macrophages especially. And this extra antioxidant in the macrophages and airway resulted in protection from cigarette smoke, from oxidative stress from cigarette smoking. And we think that was basically the mechanism for how this SNP was protective in humans. So this is our model, really. In the wild type, we have lots of SOD3 that's stuck in the tissue around the vasculature. But with the polymorphism, you lose that binding to extracellular matrix, and the protein diffuses into the extracellular space, also plasma is shown below. And that excess antioxidant enzyme in the extracellular space helps kind of protect the macrophage-induced oxidative stress that's induced by cigarette smoke. So that is the candidate gene era. And right around the time I was finishing this, the early 2000s, the whole genome was, human whole genome was sequenced, but it still wasn't practical to do that at scale. But gene arrays were practical. And I ended up working in, that was my first foray into omics, were the Affymetrix microarrays. And I published one of the very first publications and first deposits into the NCBI Geo database. But anyway, this work, which I'm not going to highlight too much, but really got us into thinking about how do we do population-based omics studies. And as you can tell, I was interested in proteins, but also genes. So I ended up starting a COPD cohort of a couple hundred patients. This morphed into larger cohorts. The idea of these cohorts were really to collect samples so that we could study DNA from patients, we could study RNA and transcription, we could study proteins, translation, and we could study small molecules or metabolites. And then look at what the association between those molecules were and phenotypes such as emphysema. And then later, as we moved on, you'll see some of this evidence. We did the GWAS studies, the transcriptomics studies, the proteomics studies, metabolomics studies, but we've begun really in the last 10 or 15 years to combine those large data sets and do QTL studies and then network studies as shown in the lower corner. That was only possible because there have been enormous leaps in technological advancement, including rapid and cheap whole genome sequencing. Now you can get a long read whole genome sequence for about $300. This is just amazing to think about that. But also epigenetics through bisulfite sequencing, or also through arrays, TAC-seq and ChIP-seq, transcriptomics that's now done through RNA-seq, for instance. And then proteomics, which is really my area of expertise. And the ways of doing proteomics are mass spec or array-based or antibody-based, as shown here. And two of the leading arrays include SomaScan and Olink. Then also metabolomics, either by LCMS or NMR. And again, as pulmonologists, we have the option of doing both lung and blood. Blood is a lot easier to get, but I think oftentimes lung is very informative. So most commonly, it's a little easier to do bronchoscopies than to get lung tissue. and then the least invasive way is maybe doing exhaled breath condensates, which we've done a little bit. Those are more challenging. I won't talk really at all about those. And then we have also, I think, been fortunate to have advancements in how we analyze these large data sets. And two of the different strategies we use are pathway enrichment. Those are using pathway enrichment tools such as Reactome, IPA, or Go. But you can also build your own networks. And that's what we're doing a lot in COPD. There's an example of one multi-omics network identifying particular proteins that we're studying, sRAGE, leptin, sphingosine kinase. These are all important proteins in COPD. I'll talk a little bit about one of them at the very end. So the biggest challenge, and this is why I want to encourage international collaborations, because the biggest challenge is getting large cohorts and data, and also diversity. So it's important to study populations across the world, not just European or US populations. So I helped put together a consortium of cohorts with lung data and proteomics, in this case, SomaScan proteomics. And these are cohorts you know a lot about. They're much larger now, but when we published our first paper, these were the sizes of them. And these cohorts had both spirometry, CTs, lung function data, Framingham, Cora, Mesa, Lovelace smokers cohort, COPD gene, spiromics, and Jackson Heart Study are just a few examples of the cohorts that we put together. And here are some of the really exciting findings we found. Not all of this is my work, but I've certainly contributed a lot to all of the slides shown coming up. This is a Manhattan plot of the different SNPs and genes that have been associated with COPD. This was a publication from about 7 years ago, showing lots of hits for genes associated with lung function, in particular COPD. I'll just point out one, the AGER, that's the codes for the RAGE protein. That came out as one hit. But we've also done transcriptomic studies, and this was a study published in COPD Gene and Eclipse, identifying lots of genes as shown in this volcano plot on the left, and then taking those genes and using pathway strategies to identify pathways associated with COPD, such as interferon alpha responses and beta-catenin signaling. And then we've used these samples to do proteomic studies. This is one that we published many years ago. And it shows a volcano plot identifying proteins associated with a COPD risk in these cohorts. And then we've also looked at disease progression, which is more challenging to study because you need multiple time points. I'll just point out some of the proteins we've identified that are biomarkers of COPD progression. And the one I'm going to focus on and finish my talk on is this protein called RAGE, or soluble RAGE. Soluble RAGE is lower in people who have more severe emphysema. And it's one of the most reproducible biomarkers in COPD. And I think reproducibility is a very important feature of a good biomarker. We also want replication across cohorts and biology that makes sense. So here's the reproducibility, 4 different platforms for sRAGE, SomaScan, an antibody platform, mass spec. They all correlate very well. Here's the replication across 4 different cohorts, COPD Gene, Eclipse, SCORE, which was a Pittsburgh COPD cohort, and spiromics. And all of them have that same relationship where the more severe the emphysema is, the lower your sRAGE is. Then the biology, you want the biology to make sense. Fortunately, AGR gene is highly expressed in the lung. It's also on immunohistochemistry. You can see it's highly expressed in lung epithelium. And then cell culture studies have shown that it's a very important in transducing DAMP signaling, as shown here. And activation of this transmembrane receptor causes activation of NF-kappa B and subsequent release of inflammatory cytokines is shown. All right, this is a mouse study I'm going to skip, but I'm just going to finish off one more time with once you identify something, how do you sort of look at it in the lab? So here's the SNP we identified. Again, this time in the CRISPR-Cas era, we found that the human genome, the human sequence was nearly identical to the mouse sequence. So using CRISPR-Cas, I made a knock-in mouse with this polymorphism, also a missense polymorphism. And we showed that it was protective of COPD in smoke models. And now we're actually developing drugs that are targeting this molecule using nanobodies and small molecule screens to try to target the RAGE transmembrane receptor to reduce the risk of COPD. I'm just going to finish off and summarize. I know it's a quick tour, but I wanted to emphasize that COPD is an example of a heterogeneous disease. We can learn a lot about this heterogeneous disease by doing multi-omics. And what we realize, similar to many complex diseases, that there's not just one pathologic molecule, that there are many different hits as shown on the genetics and also on the proteins and metabolomics, which I didn't show. There are many different molecules involved that increase the risk of COPD. Networks and pathways can help you understand that. Some of the best candidates, RAGE is an example, leptin is adipokines, another thing we're studying, alpha-1 antitrypsin, sphingolipids. These are all potential candidates, and pathways include Wnt signaling, inflammation, energy, etc. So I'm going to finish off because I'm just about at time. And I just want to, for those in the audience who are interested in learning how to do a multi-omics network and network analysis, we have an annual workshop in June that is fully paid for. It's a week-long hands-on multi-omics and network analysis workshop. I'm going to leave this on for a minute, but you can always email me. We pay for travel, hotel, food, and you do have to have some programming experience. But if you're interested in learning how to do multi-omics and network analysis, I encourage you to apply. Applications for next year have opened up and will remain open for about the next 6 weeks. So I'm going to stop here and we'll go to our next speaker.
It's great. Thank you so much, Dr. Bowler. That was wonderful. So you pay for travel from Japan too?
Unfortunately, it's just travel from the United States. But if you're from Japan and interested in participating, again, we want to encourage international collaboration. So please just email me if you're interested in participating in this workshop.
Oh, that's great. Well, thank you so much. So just kind of moving to our next speaker, we are so excited to have Dr. Edahiro, who will be presenting on he's from Osaka University, and he will be presenting on multi-layered single-cell genomics and COVID-19.
Thank you for the kind introduction, Amy. So I appreciate the opportunity to present this web seminar jointly organized by the ATS and JRS. And today I talk about how we interpret the integrated single-cell multi-omics with host genetics data, including both germline genetic variation and somatic mutation to elucidate the mechanism of underlying the COVID-19 severity. And this is the outline of today's talk. And at first, I like introducing the genome-wide association study, GBAS, and move on to the integrative analysis of the single-cell data with the germline genetic mutation and somatic mutation. As you know, this onset is influenced by both genetic and environment factors. And for the environmental factors, smoking is a representative and well-established risk factor, especially for the respiratory diseases. For the genetic factors, we can divide it roughly the two risk factor risk subtype, monogenic and polygenic risk. And the monogenic risk, the single genetic variation with a large effect is sufficient to cause the diseases. In contrast, polygenic risks arise from the combined effect of many variants, each exerting only a small or modest effect whose cumulative burden contribute to the disease onset. Today, when I talk about GWAS, I generally referring to the polygenic architecture of the disease risk. So I'd like to briefly explain what we did in GWAS. So let's focus on the GIS genetic variation with two alleles, G and A. Cases is predominantly of the C allele, while controlled predominantly A allele, suggesting that carrying the C allele is associated with increased risk of the disease onset. And look at the genetic variation with the G and C side. And the allele frequency of the G and C is comparable between case and control, indicating that this genetic variation did not associate with the disease onset. So next is about T and A. And allele frequency of A is more frequent in cases than in control, indicating that allele A is associated with increased risk of the diseases. So in GWAS, we generally perform this type of the association analysis across the entire genome, generally adjusted by the covariety such as age and sex. We calculated the effect size and the p-value of each SNP in a genome model. But the test is many, so we have to correct for the multiple testing. So we set a significant threshold like this, and we then considering the SNP that satisfy the genome-wide significant level in post-GBAS analysis. In case of the GBAS, the COVID-19 GBAS have been initiated since the early phase of the pandemic. And in particular, through the COVID-19 Host Genetic Initiative organized by the Broad Institute, through international collaboration, we conducted the large scale of the COVID-19 GBAS. This is a Manhattan plot of the COVID-19 GBAS. And upper side is the hospitalized COVID-19, and lower is the self-reported GBAS COVID-19. And COVID-19 related variants were identified in the gene region involved in antiviral response, such as interferon and chemokines, as highlighted in blue, as well as a gene lesion linked to the ILD or COPD, highlighted in the colored in the green. So GBAS is a hypothesis-free analysis, but GBAS clearly elucidated the part of the disease mechanism. The interesting part of the COVID-19 is that we expected that we should see the strong signal in the HL lesion, but in COVID-19 GBAS, the strongest signal was on the chromosome on 3. But chromosome 3 confer their allele frequency is East Asian population, so which motivated us to conducting COVID-19 GBAS in Japanese population. This is a COVID-19 GBAS in Japanese population of the young patient with severe COVID-19 and control through the collaboration with the Japan COVID-19 task force. Sample size is limited, but we identified a genetic variant which satisfied a genome-wide threshold on chromosome 5. And this variant is RS 60200309, and reference RS is G and risk RA. And odds ratio is 2, so patient with carrying the 2 risk RA, odds ratio is 4. We look closely on this loci, and we found that this variant is located in the intergenic region downstream of the DOC2. This area is East Asian specific variation, as shown here, and DOC2 has been reported to be involved in the lymphocyte activation, migration, and type 1 interferon production, motivating us to investigate this variant in more detail in post-GWAS analysis. Next is talking about a post-GWAS analysis using a single-cell data for the germline genetic variation. Again, the GWAS aim to identify the association of the genotype with phenotype by testing for the difference in the RF frequency of the genetic variant. But most disease-associated variants were alleged within non-genomic, non-coding genomic region, as I explained in the DOX variant, and their function contribution to disease still poorly understand. So it is very important to understanding the variant function, which is often called VTF in post-GWAS era. As Professor Bowler explained, the two distinct functions of the disease variant, we generally perform the molecular QTL analysis, including the expression QTL, splicing QTL, and protein QTL analysis, like that. And MQTL analysis have answered function annotation of the variant. And MQTL analysis effort initially started with the bulk RNA expression profile, as exemplified at GTEx, while current QTL catalogs are mostly at bulk resolution. The single-cell analysis have enabled to illustration a wide range of the life phenomena, and is now established itself as a key analytical tool in the field of life science. EQT using single-cell analysis enable us to capture the continuous genetic effect along the cellular state, which called the dynamic QTL, and provide a more granular insight into the molecular mechanism of the genetic variant. And there is the growing momentum for the database construction through the single-cell EQT analysis. And we constructed the multimodal single-cell immune cell atlas from the Japanese population, including a COVID-19 patient and healthy subject. And we call this cohort OASIS. OASIS contains the single-cell RNA and single-cell VD stack and whole genome and SNP genotyping, and a plasma proteomics quantified by the Olin and metagenome shotgun data. And we perform the various type of the single-cell analysis, but today I pick up the several analysis, such as the cell-type-specific EQTL and protein risk score analysis, and the projection of the various type of the somatic mutation into the single-cell space. At first, we performed the bulk RNA-seq in COVID-19 patient through the collaboration with the Japanese COVID-19 task force to reveal the function of the DOX variant. The bulk EQT analysis revealed that DOX2 was downregulated at the risk RLM, specifically in younger population. And differential expression analysis revealed that there is a negative correlation between the DOX2 expression and disease severity. And immunohistochemistry analysis revealed decreased expression of the DOX2, suggesting that this risk variant might contribute to the severity of COVID-19 by suppressing expression of DOX2. However, the bulk analysis does not allow us to resolve the cell-type-specific mechanism. So we performed the PBMC single-cell RNA analysis. And this is a UMAP embedding of the PBMC. The left side is a PBMC UMAP embedding annotation of the PBMC. And DOX2 was highly expressed in the monocyte and dendritic cell. And DOX2 expression was downregulated, specifically in myeloid cluster of COVID-19. So we focus on the myeloid cluster. This is the UMAP embedding of the myeloid cluster. And left side is the annotation of the myeloid cluster with the fine annotation. And we found that DOX2 was highly expressed in non-classical monocyte, a rare subpopulation of the monocyte. And DOX2 was the downregulating specifically in non-classical monocyte of the COVID-19 patient compared with the healthy subject. So taken together, suppression of the DOX2 expression in non-classical monocyte contribute to the COVID-19 severity. Then we performed the single-cell based EQT analysis of the DOX variant identified in the GBAS, Japanese GBAS. And we found that this variant did not affect the DOX expression in healthy subject, but we found that risk RNA showed the COVID-19 specific decreased dosage effect on the DOX expression, specifically in non-classical monocyte. So taken together, the single-cell analysis revealed that the DOX variant contribute to the COVID-19 severity via context and cell-specific EQT effect. And we also performed this single-cell based EQT analysis of the COVID-19 associated variant, identified COVID-19 GBAS and replicated in Japanese COVID-19 GBAS. Stratification by the clinical status and 6 major cell types. And heat map shows the association of the effect p-value of the EQT effect, and the heat map pattern of the COVID-19 and healthy was quite different. And the cell type which showed the EQT was different according to the cell variant. So COVID-19 associated variant had a context and cell type EQT effect. And today I will pick up the FFNR2, which is important for the type 1 interferon signal pathway. It's like a DOX2, but this IFNR2 variant also show the COVID-19 context-specific increasing dosage effect on the IFNR2 expression, specifically in monocyte. So single-cell based EQT analysis is very powerful to decipher the function of the GBAS identified variant. So far, I'm just talking about a single genetic variation. So next, I'd like to think about a polygenetic score. PRS is calculated by aggregating the effect of the common genetic variant associated with phenotypes. And PRS can predict disease risk and other phenotypic variants, and research field of the PRS is very active. However, it has not yet been investigated in detail how PRS affects intermediate trait, including transmitter and proteomics. So we constructed the hospitalized COVID-19 PRS. And using this severe COVID-19 PRS, we performed the differentially expressed and protein expression analysis across the clinical status and cell types. Differentially expressed gene and protein were observed specifically monocyte, CD8 T cell, and plasma protein from COVID-19 patient, but PRS did not affect the transcription or proteomic profile in the healthy subject, suggesting that PRS might affect the transcription and proteomic profile in a context and cell-specific manner. So last part is the iterative analysis of the single-cell data with a somatic mutation. Mosaic chromosomal alteration, MCA, is a common age-related somatic mutation in hematopoietic stem cell that create a genetically distinct blood cell population. MCA have been reported to be associated with increased risk of the hematopoietic malignancy and broad range of the benign disorder, including a cardiovascular, autoimmune, and infectious disease, including COVID-19 or COPD. And this is one of the hottest topics in the field of the human genetics. However, the underlying mechanisms of the increased risk of the benign disorder remain unknown. So defining MCA as single-cell resolution within each individual should contribute despite its biological mechanism. And this is the overview of the pipeline of the single-cell decomposition of the MCAs. We use the population scale genotype data from the Biobank Japan. The total sample size is more than 50,000, and we interpreted the BBJ genotype data and our genotype data and detect individual carrying the somatic MCA and each region MCA event occur. And utilizing the individual MCAs as a prior information enable us to more efficiently distinguish somatically mutated cell from normal cell a single-cell resolution from even spot single-cell transcript data within individual who carry the MCA event. This is the result of the single-cell decomposition and copy number alteration. And heat map shows the cell type enrichment of the CNA event. And left is the sample, and heat map shows the cell type enrichment. Not all, but several CNA events are highly cell type specific. But today I'd like to pick up the CH05 sample, which is event is occur in the chromosome 19q gain and enriched in the B cell, as shown in the UMAP in the left side, upper the right side. And luckily, this CNA event occurred in B cell, so we performed the BCR analysis, which is a distinct omics layer from genetics and RNA transcriptome data. And BCR analysis revealed that somatically mutated B cell constitute a large one BCR clone type, and interestingly composed of IgM and IgD. And we know this BCR information, so we constructed the recombinant antibody from this mutant B cell, and the recombinant antibody revealed that somatically mutated B cells show no reactivity against SARS-CoV-2 major antigen, suggesting that this MCA event have a negative impact on the acquired immunity, especially at this patient. And next is a single-cell decomposition of the loss of Y chromosome. And upper left heat map shows the cell type enrichment of this analysis, and LY cell enriched in the myeloid specific enrichment. And we perform the differential expression analysis of the LY and normal cell in COVID-19 patient of the monocyte, and upregulated deck of the LY were significantly enriched in the T-cell related pathway. And next, we perform the differential abundance analysis of the LY high patient and LY low COVID-19 patient. And this analysis revealed an increase in the myeloid cell, revealed that an increase in the myeloid cell and decrease in the naive T cell and increase in the effector T cell with LY high patient, suggesting that a monocyte with LY affect immune response via the composition change of the T cell. This kind of the observation could only be captured by a single cell interactive analysis of the single cell data and hostogenesis data. In summary, COVID-19 GBAS has reaffirmed that GBAS is one of the most powerful approaches for the dissecting the disease pathogenesis. And not only JAMA risk variant, but also somatic event converge on the specifically immune cell subset to drive functional dysregulation in severe COVID-19. And our single-cell level decomposition approach provides novel insight into how diverse genetic variation shape immune cell function. And this interactive multi-omics framework advances the mechanistic understanding of COVID-19 severity and provides a foundation for the precision genetics that can be applied to, I think, intestinal respiratory diseases. And last, I would like thanks for the Extremizing Society gratitude. And this study made possible through the contribution of the many collaborators. And I especially thanks to Professor Kumanogo, Professor Okada for their direct supervision, and Dr. Shirai for building the data set together with me. And thanks for Professor Fukunaga, Professor Ishii, and Professor Nangu for the leading the Japan COVID-19 task force. Thank you for the listening, and sorry for I can share the slideshow. Thank you.
Oh no, thank you so much. That was wonderful, and we could see it very clearly. Thank you so much. And I think it's really interesting how both speakers were kind of talking about omics and how far the technology has come, but you both kind of focused on GWAS, which is like one of the first more classical omics. So I thought that was really interesting. So we are opening it up to Q&A, and it looks like we have a question from Dr. Bowler. Do you want to ask the question, Dr. Bowler, or?
That was a very nice presentation. Thank you. Your findings, I think, really illustrate the importance of diverse populations because the DOC2 SNP mean allele
Yes.
frequency was predominantly found in Asian populations, Japanese populations, and not in European. So I think I was very happy to see that because you would have missed it if you just study European populations. I had a question about so often when we present research, much of my research is in the blood also. And sometimes people want us to study lungs and not blood. And so my question really goes back. So you studied the monocyte and DOC2 expression. What do you think, or do you know if there was any differential expression by SNP in alveolar macrophages or any lung specific cells? And how, yeah.
I'm sorry, I didn't investigate about beside a PBMC. But I think DOX2 expression is highly expressed in the immune cell, so I didn't expect that in the lung cell. But at that time, we could not access to the lung resources. So yeah, now maybe we can so human cell atlas uh, like publish the single cell data, or some group also reported that deposit the single cell equity resource of the lung. So we can check it, but we did not check that. But at that time, we cannot check that. Yeah. But GTX data, it's a bulk RNA-seq data. We cannot check that. We cannot, yeah, because of the low frequency of the IL in European. GTX is composed of the European. So in GTX, we can, yeah, find out the equity effect.
I had a related like follow-up question to that. So I guess Russ, like your focus on the focus of the talk on SRAGE and the relationship with emphysema. Have you thought about it in terms of like pulmonary rehab and if maybe it has a role too with respect to muscle?
I haven't, but I would be open to future discussions with you about that.
Yeah.
It's is found to be associated with other diseases such as Alzheimer's disease, cardiovascular disease, and certainly diabetes. I don't know about musculoskeletal diseases, but I wouldn't be surprised.
I started poking around. People have looked at it, but it's coming up in cardiac rehab, but nothing too much with pulmonary. And then I had a related, like it's kind of related, it's kind of like small world in a way, Dr. Edahiro. When I was doing my postdoc, I did this analysis and we found a genetic association to DOC1 with weight loss, like change in BMI, in cancer, and also COPD. And that one, DOC1, is actually expressed in muscle. And so I just kind of thought that was interesting that you found that association with end stage, like more severe, like COVID. And just kind of thinking about that, I started poking around, just like looking at that pathway, those DOC proteins. Like one is more immune cell, but the DOC1 that I saw before has a role, it's ubiquitous, like across tissues, but it does have a role with muscle. Like trying to understand, like maybe you're also picking up things that are extra pulmonary, even extra like inflammatory, like more like skeletal muscle, like adipose tissue, to get like to more the end stage in terms of COVID. I don't know if you've thought about that too.
Um, I'm sorry, I'm not perfectly understanding, but
Yeah, I think I spoke too
yeah,
fast.
I we don't invest about um muscle or yeah dog one we didn't
Yeah.
is yeah, but what we so today I didn't show, but we also performed the experiment using Syrian hamster, Syrian hamster.
Okay.
Yeah, and what we found is that so dogs might be involving in the migration of the immune cell to the lung inflammatory cell, because DOX2 suppression is contribute to the viral load much more, and monocyte or lymphocyte migration is decreases. So we speculate that DOX2 expression at early phase as a genotype of the risk area might be contribute to the DOX2 down regulation and contribute to the loss of the losing of the migration of the lymphocyte. Then viral load is higher, then contribute to the systemic inflammation like a cytokine storm. So we speculated that. Yeah.
Great. Thank you for
Great.
sharing that.
I think we probably have time for one more question. Dr. Gotoh, do you have any thoughts, or maybe we could think about how we can collaborate more internationally? Dr. Gotoh, do you have?
Thank you.
Yes.
The wonderful talk. Thank you very much. I would like to have a question to Dr. Russ, the talk. So wonderful talk. Thank you very much. And you showed that the S-RAGE expression is related to the degree of emphysema. So is this due to the splicing changes?
Due to what type of changes? The splicing. No, not splicing. It is a PQTL for the protein that the SNP is. Although the PQTL is independent of the relationship with, or not independent, but in people who have the wild type, so the common variant, have the strongest relationship with emphysema. There's a less strong relationship in people who have the variant. And we're working out those mechanisms with the knockout mice. It's thought to be due to binding of ligands in the extracellular receptor, but we're working on that right now.
Thank you. So regarding the collaboration between the ATS and JRS, and practically, so in our assembly of the Japanese Respiratory Society, we don't have the specific section about genome and genetics, and we need to continue to how to build some team of the interdisciplinary approach. So we'd like to discuss in the future, but we have excellent talent like Edahiro sensei and other researchers in Japan. So we'd like to continue to interact with ATS.
One way
start up with this joint symposium.
Yes. I think that is the next step, would be proposed joint symposiums. We've missed the deadline for ATS. The next deadline, it's typically in June or July. So maybe Merry-Lynn, you and I we can reach back in the next 6 months, talk about joint symposium for 2027.
Do you have a similar opportunity too through your society? Like, is there a joint, like an application that we could put in to JRS?
6:02 p.m.
What? So there's an annual JRS respiratory science meeting
In April, right?
in April. Okay. And when do those proposals are they due?
I need to discuss in the next JRS meeting. So the first program committee is held in the April.
Okay.
During the JRS conference. So we can propose some idea at that meeting.
So maybe we should do that. We could do a joint proposal. You could do it in April. We'll do in June a joint effort. for 2027.
Okay, okay.
That's great.
Thank you very much.
Yes, yeah, thank you.
Thank
Well,
you.
I think we're a little past the hour, so again, we wanted to thank so much Japanese Respiratory Society for joining us and American Thoracic Society for supporting. And we did figure out the time zone, which I'm so figuring out the time to do the webinar. I think it all worked out, so we're just really excited about that. Thank you so much to my moderators, Dr. McDonald, Dr. Gotoh, and then our speakers, Dr. Bowler and Dr. Edahiro, for that wonderful international discussion. And Daniel, thank you so much for hanging on, and Dr. Moore as well.